look at the peripheral nervous system. Last week we looked at the central nervous system. Now we'll look at things that are outside the brain and spinal cord. So the peripheral nervous system is a link to and from the outside world, the outside our body. It's the communication lines to and from the central nervous system. So it's all the neural structures outside the brain and spinal cord, and you can break those down into four parts. The sensory receptors themselves, the transmission lines, those are the nerves that carry information from receptors to the central nervous system or from the central nervous system outward. The motor endings and motor activities. We talked about skeletal muscle contractions before. We've talked about activating glands. This is how we actually do it. Actually, we're going to be talking about glands in the future. And we will also touch on reflex activity. So we have the central nervous system that we spent last week talking about, the brain, spinal cord, cranial nerves, spinal nerves all break off into and come out through the peripheral nervous system. Peripheral nervous system is immediately divided into two different groups of nerves, those that are bringing information in, the sensory or afferent nerves, and those that are taking information out, and that would be the motor or efferent division. The motor division is also divided into two groups. There's the somatic nervous system that controls skeletal muscles. When we want to contract a skeletal muscle, we use the somatic nervous system. And then there are some things that need to be automated, the autonomic nervous system. And that's going to have to be divided into two as well, because there's a sympathetic and a parasympathetic division. One division tends to turn things down, the other division tends to turn things up. So let's start with the sensory receptors and sensation. So sensory receptors are designed to respond to changes in the environment, and those changes we're going to refer to as stimuli. So activation of these sensory receptors results in a graded potential, and if enough of a graded potential is generated, you can trigger a nerve impulse. There is awareness of the stimulus, which would be the sensation part, and then there is interpretation of what that stimulus means, and we call that perception. So they're very related, but they're not quite the same, and all of that happens in the brain. Three ways to classify receptors. We can classify them by the type of stimulus they respond to. We can classify them based on their locations in the body, and we also can look at the structural complexity of the receptor. So stimulus type, we have mechanoreceptors that respond to mechanical changes, things like touch, pressure, vibration, and stretch. Thermoreceptors would be sensitive to changes in temperature. Photoreceptors are able to respond to light energy. We have those in our retina. And chemoreceptors are able to respond to chemicals. Chemicals dissolved in fluids are going to be able to give us our sense of taste, smell, but they're also used internally to keep track of blood chemistry. There are also nociceptors, which are sensitive to pain-causing stimuli. Extremes of heat or cold, excessive pressure, inflammatory chemicals trigger these nociceptors. And they are very important because they tell us to stop doing whatever it is that's stimulating them. Location, we have location of where the stimuli is coming from, really. We have exteroceptors that respond to stimuli that are rising outside the body. We have receptors in our skin for touch. We have others for pressure. All of them, if they're overstimulated, will give you pain signals, but there are also receptors for pain and temperature. Most of our special sense organs, which we will all find in our heads, sight, taste, smell, hearing, are exteroceptors. Interoceptors or visceroceptors respond to things that happen inside the body, in our viscera, in our blood vessels. They're sensitive to chemical changes, tissue stretch, temperature changes. Sometimes cause discomfort, but usually we're mostly unaware of their workings. They're providing important information to regulate homeostasis. They're providing information about our body position so we can make a plan to move where we want to move, running in the background to a great extent. Proprioceptors are responding to stretch in skeletal muscles within tendons, joints, ligaments, and connective tissue coverings. And they, again, help to inform the brain of one's movements. Majority of receptors belong to one of two categories. They are either simple receptors of the general senses or receptors for special senses. The simple receptors are modified dendritic endings of sensory neurons. They're found throughout the body and monitor most types of general sensory information. The receptors for special senses are going to be vision, hearing, equilibrium, smell, and taste. Those are all housed in sense organs. They're called the special senses, and that will be covered in Chapter 15, which at this point I would anticipate us picking up again in the fall, hopefully when things are back to normal. So general senses include tactile sensation like touch, pressure, stretch, vibration, temperature, muscle sense. There's not really one receptor, one function relationship. Most of these receptors can respond to multiple stimuli. The receptors are either going to have non-encapsulated or encapsulated endings. So non-encapsulated would be referred to as free nerve endings. The non-encapsulated one are abundant in epithelial connective tissue. Most are non-myelinated. They're of the small diameter group C fibers. Their distal terminals have a kind of a knob-like swelling to them, and they respond mostly to temperature, pain, or light touch. The thermoreceptors are activated by temperatures from 10 to 40 degrees Celsius. They're located in the superficial dermis. Their heat receptors are activated at 32 to 48. They are found deeper in the dermis. And any of these thermoreceptors that are sense temperatures of outside of those ranges will lead to nociceptors being activated and interpreting it as pain. Those pain receptors are triggered by extremes in temperatures, also pinch, and the release of chemicals from damaged tissues. One of them is a vanilloid receptor, which is a protein in the nerve membrane that's a main player in all of this. It acts as an ion channel, which is opened by heat, low pH, and chemicals. For example, capsaicin in red peppers will open this vanilloid receptor. There are also itch receptors in the dermis. They could be triggered by things such as histamine. The tactile discs, or Merkel's discs as they are traditionally named, function as light touch receptors. They're in the deeper layers of the epidermis, above the dermis. The hair follicle receptors are free nerve endings we've talked about before that wrap around hair follicles. They act as light touch receptors, help to detect bending hairs. That allows us to feel bugs landing on our skin, such as mosquitoes, which we should be heading into mosquito season very soon. So we have free nerve endings, modified free nerve endings, and hair follicle receptors located throughout the body. Encapsulated dendritic endings are almost all the mechanoreceptors. They have terminal endings are encased in connective tissue capsules. They vary greatly in shape and size. They would include Meissner's corpuscles. That's the former name. They're now called tactile corpuscles. These are small receptors that are involved in discriminating touch. They're found just below the skin, mostly in sensitive and hairless areas such as our fingertips. There are lamellar corpuscles, which are traditionally called Pacinian corpuscles, after the anatomist who described them. These are larger receptors found deeper, and they respond to pressure and vibration when it's first applied to them, and at that point they get turned off, and we'll talk about that process later on, but these are deeper in the dermis. There are bulbous corpuscles, used to be called Ruffini's endings, again after the anatomist who described them. They respond to deep and continuous pressure. They're located in the dermis. Muscle spindles are spindle-shaped proprioreceptors that respond to stretch within muscles. There is one called tendon organs, which is a proprioreceptor in tendons detecting stretch. And then there are joint kinesthetic receptors that monitor the joint position and motion. We also need to be able to process that information. We need more than just sensation. Sensation is the awareness of change in the internal and external environment, and perception is the conscious interpretation of those stimuli. So the somatosensory system is that part of the sensory system that serves the body wall and limbs. It's going to get inputs from exteroreceptors, getting information from outside the body, proprioreceptors, those information about our skeletal muscle positions, and interoreceptors, information about the internal part or the visceral part of our body. All of that input is going to be relayed toward the head. Some of it will make its way all the way up to the cerebral cortex, but whether it makes it there or not, it is being processed along the way. Some things can be done in the spinal cord, other things require further up in the central nervous system, but not everything requires a fully conscious awareness of this. So the integration of this system can be thought of at the receptor level, the circuit level, and the perceptual level. So the receptor level are the receptors we just went through. How that information makes it to the perceptual level is called the circuit level. And then the cortical areas would be the perceptual area where we have perception of what's going on. So at the receptor level, we have to generate a signal. And in order for any sensation to occur, the stimulus has to excite a receptor. It has to reach an action potential, and that action potential has to reach the central nervous system. In order for that to happen, the stimulus energy has to match the receptor specificity. You can shine all kinds of bright light on the ends of your finger, but those touch receptors do not respond to light. They only respond to touch. That stimulus has to be applied within a receptive field. So again, we can see light, but we can't see it if it's shined on the end of our finger unless we're looking at it. Transduction has to occur. The energy of that stimulus has to be converted into a graded potential, which we will call a generator potential in receptors, or a receptor potential in the special sense receptors. And then that graded potential has to reach the threshold so that it generates an action potential. Remember, graded potentials are local and action potentials are transmitted. So we talked about some of those touch receptors being stimulated and then turning off. That we refer to as adaptation. So changes in sensitivity of the presence of a constant stimulus are important for a lot of nerves. As you continually stimulate that nerve, the receptor membranes become less responsive. Their receptor potential is going to decline in frequency or stop. We have some phasic receptors, which are fast adapting. They send signals at the beginning or the end of a stimulus, and that would be those receptors for pressure, touch, and smell. And we have tonic receptors. Those are the ones that adapt slowly or not at all. An example of those would be the nociceptors and most of the proprioreceptors. I will warn you, I know there's a homework question somewhere along the way because it comes up every year about which receptors are slowest to adapt. And you got to be careful. In the answers to that question, there is a receptor that does not adapt, and that is not the right answer. So in that question, they're looking for a receptor that's slow to adapt, but does eventually adapt. At the circuit level, there are pathways of 3 neurons that conduct sensory impulses and received from receptors upward to the cortical regions. So those would be the first order, second order, and third order neurons. First order neurons conduct impulses from the receptor level to the spinal reflexes or to second order neurons in the central nervous system. Second order neurons transmit the impulses to third order sensory neurons. And those third order sensory neurons conduct the impulses from the thalamus to the somatosensory cortex, where we would have the perception of the sensation. Interpretation of that sensory input depends on the location of the target neurons in the sensory cortex. So again, we have this homunculus map of our body, and we know where information is coming from based on where in the brain it is stimulated. Some aspects of sensory perception include perceptual detection, which is the ability to detect a stimulus. That requires summation of impulses. We can also get magnitude estimation. The intensity would be coded in the frequency of impulses. And spatial discrimination. We can identify sites or patterns of the stimulus, and that would normally look at in the lab using a 2-point discrimination test. So when you read about that in this week's lab, that's what we're talking about, spatial discrimination. We can also get feature abstraction. We can identify more complex aspects of several stimulus properties. Quality discrimination, we can identify between submodalities of a sensation. Is it a sweet taste or a sour taste? Pattern recognition is going to be very important. We can recognize familiar, significant patterns, melody in a piece of music. Sometimes we can recognize patterns where they don't exist. We are so good at pattern recognition. So perception of pain is important. It warns us of an actual or impending tissue damage. It is a protective response, tells you to stop doing what you're doing. Stimuli for that would include extreme pressure and temperature, as well as histamine, potassium, ATP, acidity, and a chemical called bradykinin. Those impulses travel on fibers that release neurotransmitters glutamate and substance P. And some pain impulses are blocked by inhibitory endogenous opioids such as the endorphins. All of us perceive pain at the same stimulus intensity. So someone doesn't get more action potentials than someone else. But that our pain tolerance, our perception of pain, is different. Sensitivity to pain means a low pain tolerance, not a low pain threshold. And there are genetic reasons for that, as well as response to pain medications, and lots of research into how to control pain. We do have to talk about visceral and referred pain because we don't often get a lot of pain information from our viscera. Sometimes we do, and usually it's a vague aching feeling. They could be activated by stretch, ischemia, chemicals, muscle spasms. But what happens is, because we don't often get that information, our brain mapping is not quite as good as it is for the somatic mapping. So we end up with something called referred pain, and that's when the pain from one body region is perceived as coming from a different region. It turns out that the visceral and somatic pain fibers travel along the same nerves. And when I say nerve here, I don't mean an axon, I mean a nerve organ, so that the brain assumes that stimulus is coming from that somatic region, that common region. So for that reason, when there is pain in your heart, it is interpreted by many to be coming from the left arm. So left arm pain during a heart attack, jaw pain during a heart attack, and there are others. The lungs and the diaphragm often show up on the left side of the neck and shoulder, whereas the liver often shows up on the right side and even in the area where you might expect it from the liver. And there are a number of different places where there is a known referred pain. Long-lasting intense pain, such as a limb amputation, can lead to hyperglygia, which is a pain amplification. Chronic pain and even phantom limb pain. I think it's due to NMDA receptors that are activated by long-lasting intense pain. That allows the spinal cord to learn hyperglygia. And early pain management is critical to preventing this long-term problem. Phantom limb pain is pain that's felt in the limb that has been amputated. And the use of epidural anesthesia during surgery can help reduce phantom limb pain. One thing to be aware of is that even though that limb is gone, that part of the brain that has been receiving signals from that limb is still there. It shouldn't be receiving signals, but the information is still there. The ability to do that is still there. So that may be part of phantom limb pain. I will stop there, and then we will look at nerve transmission.